Thank you all for joining us today for the webinar. My name is Ellie Thomas, and I'll be coordinating the webinar today. I am here with my colleague Katie Maxfield, and today Katie will give a live demonstration of protein structure prediction in NovaFold. You may have noticed that your microphone has been muted. However, we do encourage you to ask questions along the way. To ask a question, just type it into the chat dialog and send to host. I will then direct these questions to Katie to be answered for the whole group later on. And with that, I will turn it over to Katie.
Thank you, Ellie, and thank you all for joining us today. As Ellie said, my name is Katie Maxfield, and today I'm going to be presenting DNASTAR's Protein Structure Prediction offering. This is part of our Lasergene Structural Biology Suite of applications. First, I'll give some background on DNASTAR and our software, and then I'll be getting into some more detailed information about our NovaFold structure prediction offering. Following this, I'll be opening the software in order to demonstrate how end users can set up and run structure predictions, analyze resulting models, and finally, review various downstream protein workflows within Protein 3D, including structural alignments and epitope prediction methods. DNASTAR has been in business for over 30 years, and we've come a long way in that time. Founded in the lab of Dr. Fred Blattner at the University of Wisconsin, DNASTAR's commercial efforts were focused on DNA sequence analysis and molecular biology software. As the fields of molecular biology, genomics, and biochemistry evolved, so did we, providing tools for next-gen sequencing analysis, and most recently, developing applications for protein analysis and structure prediction. As well, we're now offering all of our software on both local computer and cloud platforms. This long history and wide use of our applications has made DNASTAR the leader in citations in peer-reviewed journals. For the past 30 years, more researchers have cited DNASTAR software in scientific journals than any other commercially available sequence analysis software. In fact, earlier this year, we surpassed 50,000 total citations in peer-reviewed publications. We're a global software company. We're headquartered in Madison, Wisconsin. And if you have questions or require support, we have the full staff to provide guidance. And we also utilize your feedback to make our software even better. In addition, we have distributors located around the globe to supplement our in-house support team. Our software supports a wide variety of workflows across numerous platforms. Here you can see some of our molecular biology suite tools that help accelerate cloning, primer design, and sequence alignment projects. For researchers running next-gen sequencing projects, our genomics suite offers sequence assembly and alignment, expression analysis, and variant detection. And finally, our structural biology suite offers tools for protein sequence and structure analysis. This software integrates macromolecular structure visualization and analysis, and includes tools for epitope prediction and structure alignment. In addition, the Structural Biology Suite offers access to NovaFold for protein structure prediction. That's where we'll be focusing our attention today. We're currently implementing protein-protein docking workflows into this suite, but our introduction to this feature will have to wait for a later date. As a researcher, you may ask, why is protein structure prediction so important? Well, if we simply look at the number of sequence entries in UniProt, and compare that to the number of known protein structures in the PDB, we can clearly recognize a huge discrepancy. So while we're seeing a rapid growth in sequence data and gene identification, it remains difficult and costly to solve protein structures using traditional X-ray crystallography methods. Protein structure prediction programs are becoming increasingly important because they allow us to model proteins that cannot be resolved or simply have not yet been resolved. Here at DNASTAR, our development efforts are driven by quality and accuracy, and our NovaFold software is no exception. NovaFold uses the highly accurate ITASR algorithm, which was developed by Dr. Zhang at the University of Michigan. NovaFold uses a unique hybrid method to build full 3D atomic models of proteins with unknown structures. ITASR is the leading algorithm for structure prediction. For the past 10 years, it has won the CASP competition as the best algorithm for predicting unknown protein structures. A little more about the ITASR and NovaFold approach. ITASR combines both protein threading and ab initio folding. The method analyzes the sequence profile, predicted secondary structure, predicted solvent accessibility, and predicted intermolecular contacts to identify structural fragments from multiple templates. The program then constructs fragments that do not match the template and ultimately refines the model into lower energy conformations. For regions of unknown structure, NovaFold utilizes ab initio folding methods, performing calculations on the biophysical properties of the protein to generate a model. These ab initio methods work well for predicting proteins or domains under 200 residues, but the method as a whole supports predictions up to 2,000 amino acids. The image here shows an ITASR prediction on the left with a TM score of 0.818 and the experimental structure that was solved on the right. The TM score is the template modeling score. This was also developed by Dr. Zhang at the University of Michigan. This measures structural similarity of two protein models while accounting for the global fold similarity. We'll look at this metric in more detail later in the presentation, but just comparing the model to the known structure visually, you can get a sense of the accuracy of the ITASR modeling. ITASR's unique hybrid approach that we talked about allows our NovaFold software to build highly accurate models, even when there are no template structures with similar sequences available to your query. By contrast, most other applications use homology modeling methods. These methods rely solely on sequence similarity. And homology models work well when there's a high degree of sequence conservation, but they fall short when there are gaps in the protein sequences. Another powerful advantage of NovaFold is the ability to use multiple template structures during modeling. So most applications use just one template structure for modeling, but NovaFold can incorporate domains from multiple structures into a single composite model. And along those same lines, NovaFold can also create models when there are large gaps between the query sequence and the template structures. Again, this really speaks to the power of the hybrid approach of threading and ab initio folding methods. Another unique feature of NovaFold is that it performs a flexible refinement step. This looks at all atoms in the model and performs energy relaxation and minimization across the entire model, including allowing the backbone to be flexible. So all of these steps, from template selection, simulation and modeling, and model refinement, happen automatically. The end user needs only to submit the protein sequence, and our software will do all the rest. And finally, with our newest release of NovaFold, we're proud to offer NovaFold both on the DNASTAR cloud, which offers security and unlimited computing capacity through the Amazon cloud, and also locally on a Linux workstation for those who prefer to do this work on in-house hardware. NovaFold also provides flexibility to the user. As I mentioned, the software supports predictions for proteins up to 2,000 amino acids in length. And we now offer the ability for users to customize the modeling by defining known atomic distances or providing their own template structures. And when you provide your own template, you can input a structure file from your own library or provide a PDB identifier. Additionally, you have the option to provide your own sequence alignment, or you can let the software perform the sequence alignment automatically. And finally, as I mentioned, NovaFold offers the flexibility to run your structure predictions locally or on the cloud. Ease and flexibility of access is a hallmark of DNASTAR software. Our software can be run locally on a desktop, network, or workstation cluster, and we also offer unlimited use site licenses. As an alternative, we offer cloud access as well. We have clients that have large projects that are few and far between and don't want to make the investment in building infrastructure or purchasing hardware. So we give them the option to access our cloud-based tools. And then finally, DNASTAR also provides contract services for both protein structure prediction and genome assembly. Here at DNASTAR, we want to be an extension of your lab. With our contract services, we're able to tap into the expertise of our team of molecular biologists, computational biologists, computer scientists, and bioinformaticians to assist with your data analysis requirements and, most importantly, deliver quality results. So at this time, I'm going to move out of the presentation and give a live demonstration of our structure prediction tools. Before we get into the software demo, though, I'd like to open it up to any questions that have come in.
Yeah, thanks, Katie. We've actually had a few questions come in so far. And the first question is, if I wanted to run NovaFold locally, what kind of computer do I need?
Thanks, Ellie. That's a good question. So the local version of NovaFold runs on a Linux workstation. If you're considering running the local version, our technical support team can help you get that set up and provide details about any additional requirements you need on your Linux box.
Okay. And another question that came in was, how long does the average prediction take?
So that really depends on the size of the protein. For example, predictions for proteins less than 400 amino acids in length can generally be completed in a day or less. Larger proteins may take a couple of days to complete, but the important thing to note is that that prediction process is completely automated. So as an end user, all you need to do is submit your protein sequence, and NovaFold will do all the rest. So that wait time is just that, it's wait time. Once the prediction's done, you can come back and do your analysis.
Okay, thank you, Katie. And as a reminder to our audience, you can chat your questions in at any time, and then I'll ask them later on in the webinar, time permitting. And with that, I think Katie's going to get started with the software demo.
Great, thank you, Ellie. So before I jump into our structure prediction tools, I just want to point out that here I have the DNASTAR Navigator open. The Navigator helps you get started with your projects, showing all the different components of our software, organized by workflow. And as I hover over each item, you can see a short description, as well as links to videos, tutorials, and sample data. All these resources are designed to make using the software easy and to help you get to the right application for your work as quickly as possible. So from here, I've launched Protein 3D. This is our protein structure visualization and analysis application. And from here, we can open a PDB structure directly from the protein databank just by typing in the PDB identifier. You can also open structures from your own library in PDB format. And then from there, you'll notice we can visualize the structure, the sequence, and we can also look at our analysis methods to investigate the biophysical properties of the protein. But for many proteins, it isn't feasible to obtain an experimental structure. Crystallography is expensive, and beyond that, many proteins just aren't amenable to crystallization. So to address this problem, we're offering protein structure prediction in NovaFold. So to get started with my protein structure prediction, all I need to do is go to File, Predict Structure with NovaFold. And I'm going to expand this window just for clarity. And from here, I'm just going to log into my NovaFold account using my DNASTAR credentials. So now I'm logged in and I have access to my NovaFold license. And from here, I can add a sequence directly by clicking the plus button and then navigate to my sequence file in FASTA or GenBank format, or in this case, I'm using the DNASTAR protein format. So I navigate to my file, click Open, and then you see my sequences added to my prediction list. At this point, it's unsubmitted. So to submit this sequence for prediction, I'll just click the Run button. And here we're presented with a summary of the sequence that I'm submitting, including the sequence length and the actual amino acids in the sequence. You're also presented with a few advanced options. Now, generally, you can just hit Run without any additional changes here, but I just want to point out that NovaFold will perform a step to predict ligand binding sites and protein function. You can uncheck this if you want to save a little bit of time on your prediction. So in this case, I'm just going to leave all the defaults and click Run. And then you can see that that prediction starts to spin up on the cloud. So at this point, I can walk away and let NovaFold do all the rest. Even if I close out of the software or shut down my computer, the prediction will run to completion without any interaction from me. So while that's running, you'll notice that I have some other predictions listed here that have run to completion. And these will be listed on when I log into my account from any computer, so I can access them anywhere. So to get a better understanding of what the prediction results look like, we'll just open the top prediction here and look at the NovaFold report. So now I'm going to walk you through the results in this report, and then we'll look at a few downstream analysis options. So at the very top of the report, you see the name of the protein sequence I submitted, in addition to the sequence length and the amino acid sequence. Below that, we see a list of templates used to build our prediction model. So you'll remember that NovaFold uses multiple template structures to build the composite model. So the top 10 templates are listed here. However, there may be others that are not exposed in this report. If I want to look at any of these templates in more detail, I can click this link to open the structure in the PDB. We also have the option to align our models back to these template structures, which I'll demonstrate later on in the presentation. So in this template table, you'll notice we have the Z-score. This is a normalized score describing the similarity between the query sequence and the template structure. So with this score, the higher the better, and numbers greater than 1 would be significant. We can also see the threading algorithm used for each template structure. And then we can look at coverage. This is the percent of the query sequence that matches the template structure. And then percent ID is the percent of the template that was mapped to the query sequence. And then this mini-map just lets us see visually how our sequence aligned to the sequences of the template structures. So we'll scroll down through the report, and next we come to the model overview. This displays metrics for all of the predicted models at once. And here, the first thing you notice is the TM score and RMSD score. So these are predicted values for our model, comparing each model to the unknown structure. Both of these values measure atomic distances between superimposed structures. So a little bit about these scores. The RMSD value, or root mean squared deviation, is a more traditional measure of similarity between protein structures, and this looks at the average pairwise distance between residues. Because this value is based on average distances, outliers can skew the calculation. For example, a rearrangement of a terminal domain between two structures that are otherwise very similar could result in a deceptively low RMSD score. On the other hand, the TM score, or template modeling score, which was developed by the creator of ITASR, Dr. Zhang, at the University of Michigan, weighs close atom pairs between structures more heavily than more distant atom pairs. So because this is less sensitive to localized differences between proteins, the TM score is becoming one of the more accepted measures of model quality. The TM score is also normalized over the length of the protein, so the range here varies from 0 to 1, with 1 being a perfect structural match. Models with a TM score over 0.75 are considered high-quality models, and generally when we see a TM score greater than 0.5, we can infer that the overall protein fold is likely correct. So the models here are color-coded based on that TM score. Green models have a TM score greater than 0.75. Again, these are high-quality models, and these would be appropriate models for drug design, drug screening, ligand docking, and molecular replacement. And then orange models have a TM score between 0.5 and 0.75. These would be appropriate models for mutagenesis design, predicting active sites, predicting disease substitutions, as well as looking at alternative splicing patterns and domain boundaries. And finally, red models have a TM score between 0.17 and 0.5, and these may be appropriate for predicting domain boundaries. So that's the model overview, and as we scroll down further, we see more detailed information about each of the models. So first we come to Model 1, and here we see the TM score and RMSD values are repeated. For this model, we also have a mini structure view. This is a completely interactive view, so you can scroll in and out to zoom, you can rotate it, you can even spin the model, or you can open the model as its own document in Protean 3D. This allows you to perform further analysis on the predicted model. Getting back to the report, though, as we continue scrolling down, we see some additional information that's present because we left the predict ligand binding sites and protein structure, protein function option checked in that advanced options dialog. So NovaFold found similar experimental structures. Again, we can look at each of these in the PDB simply by clicking the PDB ID link. We also have a TM score and RMSD value comparing our model to these similar structures. And again, we can even do a structural alignment between our model and the found experimental structures. Next, we have predicted binding sites. Here we see the sites and ligands that are predicted, and we can check any of the ligands to view them on the structure. And each binding site also has a confidence score associated with it and a template structure that was used for the modeling. Continuing our way through the report, we see predicted enzyme function. This can be analyzed for models with TM scores greater than 0.5. During the prediction process, NovaFold uses a database of known enzymes to predict whether the query sequence has an enzymatic function. And here we can look at the EC number identifying each enzyme and look at the Brenda database entry. And then finally, at the bottom of the report for each model, we see the gene ontology information. So this allows us to look at predicted protein function, including ontology terms for molecular function, biological process, and cellular component. And then each matching term has a confidence score associated with it. So that's the detailed report for Model 1. You'll notice we have similar information presented for each of the additional models that were predicted. So you can see there's a lot of detail provided in the report. This will help you to assess the quality of the models and to get an idea of the protein's function. So next, you may be interested in some downstream analysis options. So we'll go back to the top of the report. So the first thing I want to show you here, we talked about this template alignment in the mini-map. But let's say you want to get a more detailed view of the sequence alignment between your query sequence and the template structures. So to do that, I'll just go to the Details panel here at the right and click Send Template Alignment to Megaline Pro. And once I click that, the query sequence, as well as the sequence for each of the template structures, is sent as an alignment to our Megaline Pro application. And so here you can visualize the detailed amino acid alignment between the query sequence and the template structures. And the first thing you probably notice here is this gap between our query sequence and the known templates. So to understand a little bit more about how NovaFold handles the modeling in this situation, we'll go back to Protein 3D. And again, with just a few clicks, I can perform a structural alignment now. So in this case, we see of our top 10 templates, we have actually 2 unique templates. So I'll select both of those. And then I'll click this link to align the checked template fragments to Model 1. And so that's going to perform a full atomic alignment of the 3 structures. So in the resulting alignment, we see in our Details panel, RMSD values reported. These describe the average distance between atoms in the model structure and in each fragment of the aligned template structures. Note that because of the gap in the sequence alignment, each template structure is actually split into 2 biopolymers. We can think of these as conserved domains in our model. But each of these fragments is also listed separately in the molecules view as well. So again, here's our structural alignment. To get a little bit of a better picture of this, I'm just going to color by chain. And so now our predicted model, which is listed at the top of the molecules panel and in the top of the sequence view, is colored green. And then the 2 domains in the first template are colored yellow and blue, and the domains in the second template are colored orange and purple. And so then looking at the structure, we can really see how these domains align to our model. If we rotate this around, we can see this segment here in green with no aligned template structures. And so that's part of the structure that was modeled using the ab initio folding methods we talked about. So in this case, I showed aligning the predicted model back to the template structures. But you can also perform a structural alignment to the predicted experimental structures that NovaFold locates. Or perhaps more importantly, you can align the predicted model to template structures in your own library. So to do that, you could open the model in a new document and then simply add your known structures to the document and perform the alignment that way. Another downstream analysis method you might be interested in is epitope prediction. So to do this, we'll start with the predicted model opened in a new document as we have here. And to help with our visualization, I'm going to apply a molecular surface. And just to simplify this further, I'm going to color the surface a solid gray. Okay, now we're going to apply the DNASTAR B-cell epitope prediction method. I'm going to move my analysis view down so we can look at the analysis methods at the same time as the structure. So I'll open the Methods tab and then check B-cell epitopes. And then here at the bottom of the analysis view, then we see predicted epitope regions. I'm going to select one of these regions, and then I'm going to color that pink on my structure. So here we can see the predicted B-cell epitope right on the surface of our predicted structure. And I want to point out that the epitope prediction method I showed here is sequence-based. We're currently working on a more powerful tool to predict epitopes based on the three-dimensional structure itself. If anyone wants more information about that, we're happy to follow up with you after the webinar. So that concludes my software demonstration. We've covered a lot of ground here today, so at this time, I'll take any questions that have come in.
Thanks, Katie, for the presentation. We've actually had several questions come in, so we will address some of those now. First off, in the report, why are there multiple threaders listed next to the template structures?
Thanks. That's a good question. So here I've opened up the report again just so we can look at that. So in addition to looking at multiple template structures for modeling, NovaFold also uses multiple threaders. Certain threaders will perform better for different folds or different protein domains. And so by combining both multiple templates and multiple threaders, NovaFold is able to give you the best possible composite model.
Okay, and then we had another question about the report, actually. You talked about the TM score and RMSD values. Can you explain the other values in the model overview?
Yes, so I believe that's asking about the C-score and cluster size and density score. The C-score is a confidence score, which is an internal calculation that's used by NovaFold to extrapolate these predicted TM scores and RMSD values. The C-score is a function of the predicted energy of the model, as well as the similarity of the model to the template structures. And then the cluster size is just the number of similar structures identified during the simulation process. And a larger cluster size is usually indicative of a lower energy conformation. And then density score is related to cluster size. This looks at the tightness of that cluster and the similarity of the cluster to the threading templates that were used.
How do I define my own template structures for modeling?
That's a good question. So this is one of the newer features of NovaFold, and it's actually unique to NovaFold and iTather. You can define your own template structure using the local version of NovaFold. You can input your structure directly in PDB format, or you can provide the software with the PDB identifier. And then you have the option when you provide your own template structure to also provide a sequence alignment, or you can let NovaFold do the sequence alignment automatically. So when you provide your own template structure, you'll see the template, the user template at the top of this list here. NovaFold will still look at other template structures to perform the modeling, but the user-provided template will be weighted more heavily than the other template structures. But beyond that, providing your own template structure, all of the downstream analysis and the model reporting would be the same as what we've seen here today.
Okay, great, thank you. Can features such as disulfide bonds be specified?
Yes, definitely. So this is another one of the user restraint controls that we're offering in NovaFold Local. Atomic distances within the query protein can be specified as part of your prediction setup, much like defining your own template structure.
Okay. Another question we had is, when you logged into NovaFold, it showed 4 predictions remaining. Can you explain what that means?
Sure. So I'll open up the NovaFold window again just for reference here. So in general, NovaFold licenses are set up on an annual basis, so users don't actually need to track the number of predictions they have left on their license. They can run an unlimited number of predictions throughout the duration of their license. We do have flexibility in our licensing. So for example, my license here is set up sort of as an evaluation license. So for those kinds of situations, we can set up prediction-based modeling or licensing. So I have a fixed number of predictions on this license, and the license just counts down as each prediction gets completed. But again, it's, we do have flexible licensing here, so it's not anything to be too concerned about.
Okay. And can I submit multiple predictions all at once?
You can. So when running our cloud version of NovaFold, you can submit multiple sequences for prediction at a time. NovaFold will actually complete just one prediction at a time, but you can submit your sequences as a batch, and any subsequent sequences after the first one will be queued, and those predictions will complete after the the previous prediction finishes up. So for all intensive purposes, the user can submit all their sequences and then walk away.
All right, great. One other question we had was, how long are the results stored on the cloud?
So currently the results are stored on the cloud indefinitely. This way you can access any of your prediction results for future reference. We don't want to delete users' data, so we just keep that available to our users. You do have the option in the NovaFold interface to delete any predictions you're not using any longer or don't want to see in the view.
Okay. Well, thank you, Katie. Some of the questions we received were a little bit more technical in nature. So if we weren't able to get to your question, we'll just follow up with you with a detailed response via email after the webinar. But before we wrap up, I just wanted to highlight a couple things on our website here. Within the next week, we'll have a recording of this up on our webinars page, which you can get to right here. And we will also email that recording to you in case you missed any part of today's presentation. Our next webinar, as you can see right here, is on Wednesday, September 16th. So you can register here to learn more about our molecular biology workflows, as you can see there. Also, on our webinars page, we have video recordings of past webinars on next-gen sequencing, molecular biology, and structural biology. And then if you wanted to learn more about our software, we have our video library. And here we have videos that cover many of the features Katie talked about today, and you can just learn about those in a little bit more detail. And if you have any other questions that come up later, you can contact us here with any questions. And with that, I wanted to thank you for joining us today. I hope you found this webinar helpful and informative, and have a great day. Thank you.